All right, welcome to this explainer. Right now, literally just beneath your skin, you've got trillions of microscopic autonomous units keeping you alive. It's an absolute marvel of natural engineering, and today we're zooming way in to take a tour.
So think about it. How exactly does your body pull off a million tasks at exactly the same time? From digesting that snack you just ate, to firing those lightning-fast electrical nerve messages, to continuously pumping blood. Well, the answer lies in these tiny foundational units of life. You see, every single cell in your body is essentially a bustling micrometropolis, fully equipped with highly specialized internal machinery that we call organelles.
To get a feel for how this all works, here's our itinerary for the tour. First, we'll check out the cell's dynamic borders. Second, the manufacturing hub. Third, packaging and shipping. And finally, generating cellular energy.
Okay, let's dive into District 1, the cell's dynamic borders, the gatekeepers of our micro-city.
Now, back in 1972, Singer and Nicolson proposed what's known as the Fluid Mosaic Model. And the key takeaway here is that the boundary of a cell is not some rigid static wall. Actually, it's a highly dynamic lipid bilayer. Imagine lipids with these water-loving hydrophilic heads facing outward, while their long water-fearing hydrophobic tails tuck safely into the interior. Because this whole boundary is in a quasi-fluid state, it's constantly shifting and flowing. Plus, it's setted with this beautiful mosaic of proteins. Some are buried entirely within the membrane, called integral proteins, while peripheral proteins are just attached to the surface for cell signaling. It's essentially a fluid, smart boundary that perfectly protects the inside from the total chaos outside.
Because this border is so incredibly fluid, getting molecules in and out has to be strictly regulated, and the cell does this in 3 distinct ways. First up is passive transport. This is just molecules drifting along the concentration gradient, kind of like a gentle river, through diffusion or osmosis, 0 energy required. But for molecules that can't just slip on through, we have facilitated transport. This relies on specific carrier or channel proteins. Think of things like aquaporins for water or ion channels in your muscles and nerves. And third, when the cell needs to shove molecules against the current, from a lower concentration up to a higher one, it uses active transport. This requires actual energy, ATP, to power mechanisms like the sodium-potassium pump. Sometimes they even couple molecules together, moving them in the same direction, which is symport, or in opposite directions, known as antiport. Pretty clever, right?
But what about organisms that don't have the luxury of our controlled internal environment? Unlike animal cells, bacteria, fungi, and plants face some pretty intense external and osmotic pressures. So to survive, they've evolved some serious armor. Gram-positive bacteria, for instance, build this thick, rigid cell wall made of covalently linked peptidoglycan chains right outside their single plasma membrane. Gram-negative bacteria, on the other hand, take a totally different architectural approach. They sandwich a much thinner cell wall between a dual plasma membrane system. This rigid setup doesn't just give the cell its shape, it's absolutely crucial for preventing the organism from just bursting like a balloon under osmotic stress.
Moving right along to District 2, the manufacturing hub, where we synthesize the city's goods.
Right at the beating heart of this manufacturing hub are the ribosomes. They were first spotted under an electron microscope back in 1955 by George Palais, and these fascinating, membraneless little organelles operate as the city's dedicated protein synthesizing factories. You'll find them scattered throughout the cytoplasm in both prokaryotes and eukaryotes, just tirelessly reading genetic instructions and assembling the complex proteins that will literally build and operate your entire body.
Now, these ribosomes aren't all just floating around aimlessly. A ton of them heavily stud the endoplasmic reticulum, or the ER for short. The ER is this massive dynamic network of membrane-enclosed tubules and flattened sacs called cisternae. When it's covered in ribosomes, we call it the rough ER, and that side of town is heavily dedicated to crafting those proteins we just mentioned. But there is also the smooth ER, which doesn't have ribosomes on its surface. Why do we need that? Well, because lipids are hydrophobic, they're afraid of water. They can't be made in the watery cytosol of the cell. So the smooth ER acts as a highly specialized facility where those lipids are meticulously crafted before they're shipped out.
Which brings us perfectly to District 3, packaging and shipping, our cellular logistics center.
If we track a molecular shipment step by step, you see an incredibly precise system at work. First off, materials are synthesized over in the ER. Second, they bud off into these tiny little transport vesicles. You can think of these as the internal delivery vans of the cell. Third, these vans travel straight to the Golgi apparatus. They dock at what's called the sys face, the receiving department nearest the ER, and pass through a series of stacked, flattened pouches where the goods are further modified, tagged, and carefully packaged. Finally, they hit the trans face, which is the shipping department. Here, the finished materials are pinched off into secretory vesicles, which travel straight to the plasma membrane to release their contents outside the cell. Honestly, it is a flawlessly coordinated supply chain.
Okay, moving to our final district, generating cellular energy, powering the metropolis.
Obviously, absolutely none of this complex cellular machinery operates without a massive amount of power. Enter the mitochondria. Sure, from the outside, they might just look like simple sausage-shaped cylinders, but their internal architecture is a literal masterclass in biological efficiency. They have a double membrane structure. The outer membrane is totally smooth, but that inner membrane, it features these deep infoldings called cristae, and those folds are definitely not random. They massively increase the internal surface area available to the cell. And in biology, more surface area means more room for molecular machinery dedicated solely to churning out ATP. That is the vital energy currency that powers literally every single action in your body.
Now, while almost all eukaryotic cells rely on mitochondria for their power grid, plant cells actually evolved a really unique alternative: plastids. If you think of mitochondria as these internal combustion engines burning chemical fuel, chloroplasts are like the ultimate cellular solar panels. They contain chlorophyll pigments safely tucked within their thylakoid membranes, and by using highly specialized light-harvesting proteins and electron transport chains, they actually capture light to drive the primary events of photosynthesis. It's an entirely different way to power the city.
So let's just zoom back out for a second. Your body isn't really just one single entity, is it? It's a vast, sprawling universe made up of trillions of these perfectly coordinated microscopic cities, all working tirelessly right this very second. The borders are being monitored, factories are pumping out proteins, logistics centers are shipping vital materials, and power plants are generating ATP, all just to sustain your existence. With trillions of autonomous little units acting in perfect unison to keep you alive right now, well, it kind of begs the question, who's really in charge?
Thank you for watching. See you in the next one. Stay tuned.